I've chosen to investigate is the presence of bacteria found in store-bought powdered infant milk formula. Some information about myself is that my name is Makenzie Durham, and I'm a senior at Tunstall High School. I will be attending SOPO School of Health Professions in the spring of 2025, where I'll be pursuing a degree in diagnostic medical sonography. One reason I chose this topic is because of my interest in healthcare and the study of illnesses. Additionally, the recalls that were brought to light during the United States powdered infant formula shortage causing fatal neonatal infections. Lastly, the gaps in regulation and limited testing of the powdered infant formula during and after production that would become sold to the public. Recalls began in 2020, presenting the risk of infection that comes along when consuming powdered infant formula due to the evidence of bacterial contamination after being sold to the public. There's been a high mortality rate in numerous infants that contract the fatal infections over the years after having consumed powdered infant formula. The main infection is now called the Chronobacter infection, due to the type of bacteria being found within the dry formula. This usually affects infants under the ages of 12 months old. The Chronobacter germs cause life-threatening blood infections that make their way into the brain or spinal fluid. Even though this invasive infection is very rare, at least 4 cases are reported each year, not including the small illnesses also found in the contaminated formula. The bacteria discovered is known as Enterobacteriaceae, and is a large family of gram-negative rod bacteria species that are present in dehydrated powdered infant formula after handling. Many of these different species are considered opportunistic pathogens that are able to survive in dry powders for an extended period of time, causing infections in consumers. This bacteria can easily survive in the infant formula because of its ability to withstand certain heat stress temperatures. This is known as thermotolerance. By knowing the powdered infant formula conforms with microbiological regulations of the Food and Drug Administration, it is still found to be a non-sterile product because of the contamination risk, but is sold to the general public anyway. The powdered infant milk formulas that produce the dry formula is unaware of the high risk of contamination that can occur during production and preparation before consumption. In processing facilities, contamination can happen if the manufacturer uses contaminated ingredients or the formula touches contaminated surfaces, such as polluted water or the exposure to heavy metal containers. Not only that, but the residue and moisture in the surrounding atmosphere of the factories and transportation units help the powdered infant formula contract bacteria. Once the dry powder is in a home or NICU environment, it can become contaminated because of the poor handling by the caretakers that are unaware of the safety measures to be taken when rehydrating and storing the formula for infants. The GAP is a type of bacteria called C. seziki that is part of the Enterobacteriaceae family that is a germ found naturally in the environment but mostly in dry powdered foods. It is an emerging pathogen that can be found in powdered infant formula after dehydration, rehydration, and storage. This study will contribute to the developing understanding and awareness of bacterial contamination within specific store-bought powdered infant formulas that are regularly supplied to the public. Observing various types of bacteria within the powdered infant formula will draw attention to the bacterial contamination, reduction in handling, and risk for infection to find effective treatment. My research question is, what type of bacteria is present in store-bought powdered infant milk formula? My null hypothesis is that if the powdered infant milk formula brands are left to incubate for 37 degrees Celsius for 48 hours, then there would be no difference between the formula types and the number of gram-negative bacteria identified. The research hypothesis is that if the powdered infant milk formula brands are left to incubate for 37 degrees Celsius for 48 hours, then there would be a difference between the formula types and the number of gram-negative bacteria identified. I chose this research hypothesis because of the literature studies that I had read presenting the specific type of gram-negative raw bacteria growth found within store-bought powdered infant formula and suggested further research on the exact types grown. The independent variable for this experiment is the type of infant store-bought powders, which are Similac, Gerber, and Infamil. The dependent variable of this experiment is the bacteria grown from the formulas. The control in my experiment is a distilled water without any powdered infant formula added. The constants in this experiment are incubation time and temperature, amount of water, amount of formula used, the disinfected workspace, temperature boiled of the water, and the infant formulas. First, I gathered my materials, which included 3 store-bought powdered infant formulas, 40 nutrient agar dishes, 40 sterile swabs, distilled water, Bunsen burner, inoculation loop, microscope, and the 4 gram staining bottles that are crystals violet, grams iodine, ethanol, and xanafran. My sample size for this experiment is 40, and the population is store-bought powdered infant formulas. After I gather my materials, I then use a hot plate on 100 degrees Celsius and boil distilled water. Once done boiling, I mix the 3 types of formulas into the distilled water except for the control. Then I streak the nutrient dishes with sterile swabs for all 40 of my samples and set the dishes in the incubator for 48 hours at 37 degrees Celsius. After completing incubation, I check for growth by counting colonies. I had gram stained the certain bacteria grown from each group that looked dissimilar by drying it onto a slide with a Bunsen burner. Then flooding each of the slides with the 4 staining materials: crystal violet, grams iodine, sanifran, ethanol, and flooded each with distilled water in between. Following that, I viewed the slides of bacteria under a high-powered picture microscope that allowed me to take pictures of the bacteria grown and stained. After completing my experiment, I compiled all my raw data within a spreadsheet, and then I conducted my statistical analysis. This dot plot shows the number of bacterial colonies grown in each of the different powdered infant formula types in the control. In this graph, there is a huge outlier of the 1,444 colonies that appeared within one of the Infamil powdered infant formula petri dishes. This difference in the colony count on some of the petri dishes was most likely due to contamination within the lab. It caused the mean, standard deviation, and all the error bars to change within the graph, making most hard to read and see the colony numbers. This dot plot shows the number of bacterial colonies grown in each of the different powdered infant formula types without the huge outlier. The means in all the colonies in all the formulas are mostly between 2 and 4, except for the mean of the Gerber formula being 12.7. This means the Gerber showed the highest count of colonies on this graph, and the lowest was the distilled water control group that was supposed to grow no colonies. However, the distilled water did grow some colonies on a few plates that could have been caused by contamination. The error bars all overlap on this graph, presenting that the p-value is greater than 0.05, so the difference is not statistically significant. This bar graph shows the number of red gram-negative bacteria or purple gram-positive bacteria that grew within the control in the different types of store-bought powdered infant formulas. For the purpose of classifying each group, I selected a certain number of petri dishes that seemed to be unique. So the sample size for this graph was 10. I limited my search for bacteria in this study to gram-negative species that would exhibit Enterobacteriaceae, which typically grows in the formulas. However, I discovered a lot of gram-positive bacteria in these particularly odd-looking petri dishes that I selected. It showed that there are more gram-negative bacteria that grew within the powdered infant formulas, mostly in the Infamil and Similac, but odd that all the types grew gram-positive bacteria. Another abnormality on this graph is that the distilled water had both gram-positive and gram-negative bacteria. This could have been caused by a variety of things or just general contamination during my lab experiment. The statistical test I used for my data was the chi-square goodness of fit test. I used the chi-square because of my 2 categorical variables which had no bearing on one another. At the significance level of 0.05, I failed to reject my null hypothesis with a p-value of 0.96. The pictures to the right show my results of the purple gram-positive bacteria grown, as well as the red gram-negative raw bacteria in the powdered infant formulas. There is enough sufficient evidence at the 0.05 significance level to conclude that there is a difference between formula types when measuring the number of gram-negative bacteria identified. Therefore, store-bought powdered infant formula brands have different types of bacteria present. My results suggest that different brands of store-bought powdered infant formula grew both gram-negative and gram-positive bacteria. Similar to my findings, a study conducted in 2017 observed that there's a larger concentration of different types of bacteria present in the infant formulas starting at the production facility and after being used by consumers. This suggests that the dissimilar bacterial growth types could have been caused by contamination during production or my lab experiment. These findings help caretakers, NICU workers, and mothers gather an understanding and risk associated with the carelessness, unsafe handling of store-bought powdered infant formula. The limitations to my study include the lack of access to materials that would show the exact type of Enterobacteriaceae within each of the powdered infant milk formulas, as well as a small population size, as I could only choose and gather a few brands of store-bought powdered infant milk formula that are sold in the United States. Further research is needed on this topic to identify the exact types of gram-negative Enterobacteriaceae that grew within the powdered infant milk formulas, creating infections in newborns. This would offer evidence that emphasizes the growing knowledge and awareness of bacterial contamination, in particular store-bought powdered infant formulas that are regularly provided to the general public. There was a lot of trial and error throughout my experiment, from choosing a topic to developing the methods. I learned even when I make mistakes throughout this research and experimentation process, I can overcome it by trying again. And with each mistake and failure, I continue to improve my experiment along the way. I would like to give thanks to everyone who helped make this research possible. I would like to thank Mrs. Bowman for approving my experiment. I would also like to thank Mrs. Dillon and Ms. Bain for helping me with the gram staining and using the high-powered picture microscope during my experiment. Most importantly, I would like to thank Ms. Long for her assistance in developing the research and statistics behind my topic. Are there any questions? Yes, Mr. Taylor.
So in any of your research, did you come across any testing methods or testing protocols of these manufacturing facilities before they ship out their infant powder?
Mr. Taylor asked, did I come across any of the factory's shipping methods? And I learned that they used metal containers when they put them in the truckloads to deliver them to different stores, which could have also caused the contamination because of the stuff in them heavy metal containers.